Welcome to your first unexpected and fully vodcasted lecture. It's kind of like a surprise party vodcast, as I didn't really know that I would be needing to do this today. So I thank you for your patience, and I hope you get jazzed about it and enjoy this format. It's kind of like an upside-down classroom, if you will. I have a few announcements for you today. First off, I will be working through the grading of homework 1, and I hope to have them done and completed by the end of the week. I'll do my best to do so, but I do like to take my time and make sure that I'm giving credence to each and every one of your discussions regarding the ethics or philosophical questions or things that surround genetic engineering. On Friday, we'll just totally get stoked and play a game again. So we'll come back and finish our macromolecule game, and so it'll just be a Friday full of games. And today is the last day to enter your pre-test and knowledge surveys. I noticed that most of you have already done so, but if you haven't yet had a chance to do that, you'll want to go ahead and get that done. Homework 2 is posted, and again, as is always going to be the case, it'll be due on Tuesday. So just keep watching those Tuesday due dates, and every Tuesday you'll have something that you want to submit. With your poster projects, I'd like to be making continual progress, and I know that there are a couple of groups that have already talked with me about topics, and there's some awesome topics already milling around in your mind. But if you could please formally email me or talk with me in a formal way about what your topic will be for your group project, that would be absolutely fantastic. I'm looking forward to some super topics. And hey, if you're at a point where maybe you kind of have a few that you're mulling over and you don't know which one to choose, maybe come and talk to me about that. I don't need for this to be like completely finalized this instant. I just want you to be thinking about it and to really be coming towards a narrowing, a funneling in your focus, and hopefully starting to choose something that is a specific topic. For lab, tomorrow you'll have your pre-lab 1 due, so don't forget to hop into the Canvas shell for lab and get that submitted, and then we'll be all ready to go to do some microscopy and talk about the ubiquity of microbes in lab on Thursday. So I'm totally getting excited for that, and I hope that you are as well. That being said, we're ready to jump on in, and today we're going to be talking about the prokaryotic cell. But before we can get to looking at cellular structure, function, and all of the nitty-gritty there, we really need to take a broad view, a broad picture look at the domains of life. So I want to start with a quick reminder. Recall that on the very first day of lecture, we talked about the fact that we can split the study of microbiology into 2 sections. We can talk about both life forms and agents. Remember that the life forms are cellular. They're considered living, and they encompass what we'll talk about here momentarily: bacteria, archaea, and eukarya. So these are the life forms. We can also talk about the study of agents. So if these are the life forms that we're about to mention, recall that the other thing that we talk about and study in micro are the agents. And remember that the agents are considered acellular, they're considered non-living, and those encompass things like viruses, viroids, satellites, and the epic prions. So that being said, and situating ourselves here within the study of life forms, we can split organisms into 3 domains. And of course, we're encompassed in this, in the domain eukarya. Also, though, there are microorganisms that get placed firmly in the domain eukarya, and those microorganisms include the fungus among us, so fungi, but it also includes the protists. And the protists include what is still called, and although debatable whether this deserves a term, the protozoa. Tend to be chemoheterotrophic, that is, eating other things, versus the algae, which are self-producers, photosynthetic. So we will talk more about that at a later time. But I want to note that there are certainly microbial members of the domain eukarya, and we can split that. That is one of the 3 domains. The other domains are the archaea and the bacteria. Now, the archaea in their own right are organisms with unique characteristics. In fact, we sometimes tend to lump them into a group that we call extremophilic organisms, although that doesn't describe every member of the domain archaea. However, they do often tend to like crazy places, at least as defined by us as crazy. They like cold temperatures, hot temperatures, acid, things like that, that maybe we look on as being inhospitable inhospitable environments. So the bacteria, the archaea, and the eukarya, these are our three domains of life. And I want maybe to convince you today that although we tend to focus on the eukarya because we are, of course, members of that domain, in fact, it turns out that we are not the dominant life forms on Earth. And in fact, I think some of you will be super jazzed to hear that I'm going to read to you today. And I know that both Julian and Ashley said they really like to read, and Graham too. And so I'm going to read to you a passage from a particular author that I hope that you're going to get stoked about and start doing more reading of Stephen Jay Gould if you haven't already. So let me read you this sexy passage from a work by Stephen Jay Gould, Planet of the Bacteria, written in 1996. And this passage is so sexy. So on any possible, reasonable, or fair criterion, bacteria are and always have been the dominant form of life on Earth. Our failure to grasp this most evident of biological facts arises in part from the blindness of our arrogance, but also in large measure as an effect of scale. We are so accustomed to viewing phenomena of our own scale, sizes measured in feet, ages in decades, as typical of nature. Individual bacteria lie beneath our vision and may live no longer than the time it takes me to eat lunch or my grandfather to smoke his evening cigar. And then, who knows, to a bacterium, human bodies might appear as widely dispersed, effectively eternal, or at least geological, massive mountains fit for all forms of exploitation and fraught with little danger. So, awesome passage from Stephen Jay Gould, convincing us that yes, in fact, bacteria, and not us, are the dominant life forms on Earth. Now, let's talk about something else kind of contentious. That is to say that, in fact, archaea and bacteria have typically been lumped into a group that we call the prokaryotes. However, you'll notice that I put a question mark, a big question mark on prokaryotes, because really, does that term need to exist? Archaea, bacteria, and eukarya are all domains of their own right. Archaea share some features with eukarya, they share some features with bacteria. They are unique. And so really the grouping prokaryotes is another way to say not one of us or not eukaryotic. So we can ponder throughout the rest of the semester whether you think that the term prokaryote should be retained. We still do use it. But we might argue that it might be better to simply refer to the three domains of life: bacteria, archaea, and eukarya. Now, briefly, I also want to make another note, and that is that the spelling of prokaryotes is variable. You might at some point watch me write on the board, and I'll spell prokaryotes with a C instead of a K. It's not because I am spelling challenged, although that might very well be true, but it is because, in fact, there's multiple spellings for prokaryotes, and either one of those two spellings is totally accurate. Excellent. Let's move ahead because we need to talk nomenclature. That is naming. And so when we talk about these life forms, we need to think about how we give name to these life forms. And just like you and I, bacteria and archaea and many eukaryotic microorganisms also have a first and a last name. That is to say, they have a binomial system of nomenclature, two names. The two names are always underlined or italicized. So no matter what, when you write a scientific name, make sure that you get your italics feature in your computer and make sure to italicize or underline. We have two different names. The first name is the genus name. Escherichia is a good example of a genus name. The second name, coli, is the species name. The genus name is often historically comes from the name of the discoverer. So you might have wondered, well, what is this crazy Escherichia anyway? Well, a man named Theodore Escherich is who actually discovered this bacterium, so it actually is named after him. However, the species name, coli, usually derives from the location in which that bacterium was isolated. So coli, you might guess it, you bet, is isolated from the colon. So a colon-derived normal flora, generally normal flora bacterium, and that gives us a little bit of an indicator of the location from where that organism was originally isolated. One thing that you may also note is you might think, man, I've never heard anybody call it Escherichia coli, but I sure have heard of E. coli. Well, once we've used the genus name once, we can begin to abbreviate it. So after using the genus name the first time, we can go ahead and just write it as a large E and a period. So that is the common way to abbreviate it once it's been used once. However, you still want to italicize or underline the name. Now, you might also have been thinking, E. coli, ooh, that's a gnarly pathogen, right? But in fact, it turns out that most E. coli is actually non-pathogenic. It's actually part of our microbiome, and it lines the gut, actually, of every warm-blooded animal. So most strains of E. coli are non-pathogenic. So not only do we give a first and a last name to bacteria, but we also designate their strain. So after giving the species name, we'll generally follow that with something that tells us what strain it is. A strain that you may have heard of is the epic strain 0157H7. That one truly is pathogenic. So that's not one that we work with in our lab. It is a BSL-3 organism. That means biosafety level 3, meaning that it is in fact fairly dangerous to work with and not something we work with in our lab. However, many strains of E. coli are simply laboratory strains. For example, E. coli B and E. coli K12 are non-pathogenic laboratory strains. In fact, you could probably lick a plate of E. coli K12 and not be any the wiser. I'm not recommending it, so don't go licking your plates next time, but certainly is not a pathogenic strain of E. coli. So sometimes strain designation is equally as important as the first and last name, genus, and species. On your homework and on the exam, I'm going to give you some practice on how to work with this binomial system of nomenclature, but please do remember that you always italicize or underline, and that when you write the genus name for the first time, you want to completely and fully write it out. This leaves us ready to talk about the actual, finally, at long last, the actual prokaryotic cell. And I want to begin by looking at possible shapes and arrangements of prokaryotic cells that we see, or maybe we should rightly say bacteria and archaeal cell shapes and arrangements. Let's begin by differentiating the two main shapes. So we can get spheres, right? And we can get rods. Oh, I've definitely got to have some giant stuffed microbe up here to show you, I'm sure, that will exemplify these. So, hey, let's find, this right here, good old Escherichia. This is E. coli. It definitely is a rod-shaped bacterium. However, if we talk about, say, something like Staphylococcus or Streptococcus is a coccus, and in this particular stuffed microbe, they're showing two cocci attached to one another. So there's one coccus here, one coccus there. So it's a little deceiving on that particular stuffed microbe. So it's a sphere. So coccus, cocci are plural. There are many cocci, whereas a coccus is singular. Bacilli, these are rods. Bacillus is a single rod. Within the world of cocci, we totally have arrangements. So you noticed that with this particular Streptococcus, that it is shown in this giant stuffed microbe as being a diplococcus. That is, there are two cocci attached to one another. In actuality, Streptococcus often forms a long chain of cocci with many of these attached to one another. So diplo, two. Often this is indicative of, well, one of the genera that forms this is Neisseria gonorrhea, right, that causes the STD, or Neisseria meningitidis, that causes meningitis. Streptococcus is going to be a good example of something that often forms the chains. So I always joke when I wear my pearls that I'm wearing my Streptococcus necklace because they're long chains. So it's of note that sometimes this long chained arrangement is called a strepto arrangement, even though in fact there are other genera besides Streptococcus that form this arrangement. For example, Enterococcus and Lactococcus, they can form these chains as well. Notice that the next arrangement occurs when organisms divide randomly in many planes. So this contrasts with the quote-unquote strepto arrangement in that is when the cells divide repeatedly in the same plane, it gives rise to a chain. But when they divide randomly in many planes, it gives rise to a big grape-like cluster. And in fact, that's the name that's sometimes given to this arrangement, is a grape-like cluster. Another name that is sometimes given to it is staphylo. So that's because the quintessential example of the grape-like cluster arrangement is Staphylococcus. So if you've heard of staph infections, they're caused by Staphylococcus, and that particular genus forms these grape-like clusters. But do take note that it's not the only genus that does that. Micrococcus can also form grape-like clusters. Also, Micrococcus sometimes likes to form packets of 4. So I feel another giant stuffed microbe coming on. Right on. These are some of my favorite giant stuffed microbes because these were actually handmade by some of my students. So we got a total tetrad going on, right? Yeah, that's pretty kinky. So tetrad arrangement there, having 4 of them, right? And that is a different kind of arrangement. One of the genera that tends to form that tetrad is Micrococcus, but also some lactic acid bacteria. For example, a radly named one called Pediococcus acetylestacea is actually forming a tetrad arrangement quite frequently. Now, if we get a complete packet of 8, so if we had 2 of those packets of 4, that one would be called Sarcina, because the name of the genus Sarcina is very representative of this particular type of arrangement. Within the world of bacilli, sadly, we really don't get much in the way of arrangements. That is to say, they're more often very random. But what is cool about the bacilli is the way they tend to differ in their shapes. So we can get coccobacilli, meaning that they kind of look like a coccus, kind of look like a rod, they're somewhere in between. We can get vibrios, which are half-moon shaped cells, and Vibrio cholerae, which causes cholera, is one example of that. We get spiral-shaped cells. A lot of photosynthetic aquatic bacteria actually are spirals. And we can get spirochetes. Spirochetes, these are rad. They're corkscrew-shaped. So hang on a minute. Man, I got a lot of giant stuffed microbes. Spec this. This is a spirochete. It's shaped like a corkscrew. And guess what? This is also syphilis. So this is Treponema pallidum, the causative agent of syphilis. So what we recognize is diverse shapes and diverse arrangements. And we can also make note that, in fact, not everything fits into these basic categories of shape. I was thinking, actually, Graham would be interested with your passion for science fiction. It turns out that there are some bacteria and archaea that are actually pleomorphic. Pleomorphic quite literally means shape shifter. So I know there's got to be some sci-fi out there where there's changelings that can shift their overall shape. That is what's going on with these particular bacteria archaea, shape shifters. Some bacteria and archaea also have really weird shapes, like they might be shaped like even squares, or sometimes they may be like cuboidal and things like that. So they may sometimes have surface extensions, that is, they may form like a long, almost like star-like appendage, and those are called processia. So the appendages that form are called processia. Lots of diversity in cell shape and arrangement. Let's think, though, about what about cell size? Maybe that's something that kind of got in your mind. How big are these? I mean, aren't they super, super tiny? Well, in general, we could say that bacteria and archaea tend to be smaller than eukaryotes, though there are some, like every rule, there are exceptions to that particular rule as well. So bacteria and archaea cells, they may vary greatly in size. They may range from 0.3 to 2 micrometers in diameter. They're often, as I said, smaller than eukaryotic cells, but that's not always the case. Look at this left-hand picture. This is Thiomargarita namibiensis. Isn't that the sexiest named bacteria you've ever heard of? But notice that some of you will recognize that thio actually means sulfur. And so these internal granules that you see inside of these cocci, very large cocci, by the way, are actually sulfur granules. And then, hey, look, namibiensis, remember that species name tends to come from where this organism was isolated. And in 1997, this was isolated off the coast of Namibia. And that's where it gets that name. It is huge. That is, we could see this with the naked eye. It's almost the size of a period on a page. So it breaks the rules of the typical size of quote-unquote prokaryotic life. Very, very large. So there are also some eukaryotic cells that are very, very small. So while we can try to make generalizations, it may be that it will be that there's always things that break the mold. So how do bacteria reproduce? That is when they want to replicate. How does that happen? They use something called binary fission. That is to say that literally one cell elongates, doubles its contents, and splits in two. So let's look at a TeacherTube video that you can totally find on YouTube. This TeacherTube video shows the process of binary fission, and we're going to watch, and I'll try to narrate as we see bacteria doubling their contents, splitting in two, and undergoing binary fission. So here we've got it. Bacteria reproducing, doubling their contents, splitting in two. And if you just start from a single bacterium and it divides every 20 minutes, it'll make 5,000 billion bacteria in a single day. Rad.